welcome to this module. This module is the end of our understanding about how to fabricate biochip using MEMS based technology. When I say MEMS, it is micro electro-mechanical systems, right. In previous modules, what we have seen, how can we fabricate this chip. In this module, let us see how can we use this chip and how can we place this chip in a mechanical casing. And that mechanical casing is we have fabricated that mechanical casing using 3D printing. Since 3D printing is also part of this particular lectures or course, I am not going to tell more about 3D printing in this particular module, but we will discuss more about how exactly 3D printing works, what are the kind of 3D printers, and then we will actually see how can we operate 3D printer in the laboratory. So, if you see the slide, what we have seen, there is a heater, and over the heater there is an insulator, over that there is an electrode, over electrode there is an piezoresistor sensors, on that there is an insulator, on that there is a gold pad, over gold pad there are SU 8 pillars, and we are making SU 8 pillar conductive. On the back side there is a diaphragm, this diaphragm is using bulk micro machining. Finally, you have this chip. Once you have this chip, then you have to place this chip in this particular 3D printed module. I will show you the blown up diagram of this module in my of this particular mechanical casing in my next lecture. Right now, let us understand how can we use this chip to understand the change in the tissue properties. So, this chip is placed here, all right, and there are press fit contacts. What is it? Press fit contacts. The advantage of press fit contacts over soldering and wire bonding is that you can easily load and unload the chip into the mechanical casing. Then what you see here is a micro manipulator. Now, this section the representation is such that we are using piezo actuator, but you can use micro manipulator, manipulator. This will press the chip with micron precision. Now, when I say pressing the chip, that is, it will move in x, y and z, in micron micrometer range with micrometer range precision. Okay. So, how we will take advantage of this thing? Now, if you see this particular section is like a funnel, So, I will place the chip, there is a chip in the bottom, right, and through this funnel like this, and all right, and this is our chip. Through this funnel we will load the tissue. So, let me just draw different color so that you can understand what exactly I mean. You see, you have a funnel, there is a chip at the end of the funnel. This chip is the one that we have fabricated, all right, and you are loading a tissue. You are loading the tissue such that the bottom of the tissue will contact the chip. This is your, let us say if it is breast cancer, is a breast tissue from breast cancer patient. Tissue from breast cancer patient. Now, what you will do? If I place the tissue in this particular funnel, what is my idea? My idea is that if I press the tissue if I press the tissue, what is on the chip? This is a chip. What is there on the chip? The chip is having a piezoresistive sensors, right. So, if I apply force to this tissue, this force will be transmitted on the chip, which has piezoresistive sensors, and based on the elasticity of the tissue, based on the elasticity of tissue, based on the elasticity of the tissue, the amount of force applied and the amount of force measured will be different, right. If I have few millinewton of force, how much force is actually transmitted to the chip or to the piezoresistive sensor will depend on the elasticity of the tissue, correct. Second, so how we will apply force? I will apply force to the tissue using indenter. This guy is called indenter. Is like this and is connected to the piezo actuator. This is a piezo actuator. Piezo actuator, indenter, piezo actuator. This is I indenter, and at the tip of the indenter, if I have, so right now we are pressing the tissue with the help of indenter, that is one thing, right. Now, if at the tip of the indenter, if I have thermal sensors or thermistor is used to measure the temperature. Okay. Now, my chip is indicated with what? Is indicated with micro heater. So, if I apply at the bottom of the tissue, let us say 37 degree centigrade, I can see what is the temperature at the top of the tissue with the help of thermistor, right. So, I said T2, this is T1. If T1 is 37 degree centigrade, what is the temperature of the T2? So, if I apply or heat the tissue from the bottom, what is the temperature at the top of the tissue? If I want to measure, or if I measure temperature T2 and T1, what I can find? I can find thermal conductivity. Thermal conductivity. Okay. Third thing, if I attach a electrode at the bottom of the indenter, electrode, okay, what is there on the chip? There is a SU8 pillars with a gold pad. So, if I apply voltage here, this one potential difference between 2 points. So, point 1 for applying a voltage is here, point 2 is my electrode. Then if I apply a voltage, so this is the indenter, right. If I apply voltage here V1 across the tissue and the chip, it's too complicated, let me draw it easy, let me just rub it down the unnecessary things. So, it becomes little bit easier to understand. Okay. If I apply a voltage between the point P1 and point P2, that is P1 is on the chip here, P1, and P2 is somewhere on the indenter, alright. Indenter has what? Point P2, that is your electrode. Chip has what? P1, which again is a contact to the gold pad. So, if there is a voltage and if there is a tissue in between, tissue would have certain resistance, right. It would have resistance. So, now if the tissue has a resistance, if I am applying voltage, then what will I find? I will find different, I will find the change in current depending on what is the resistance of the tissue, right. So, what we understand that we can measure with this technique, we can measure the mechanical property electrical property and thermal property of a tissue. Electrical property, mechanical property and thermal property of a tissue. When I say mechanical, what I mean? I mean elasticity, or you can say stiffness. If I say electrical, what does I mean? I mean resistance. If I say thermal, I see thermal conductivity. So, I can understand the mechanical property, that is elasticity or stiffness, electrical property, that is resistance, and thermal property like thermal conductivity from a single tissue, obtained from or obtained by biopsy, obtained using biopsy, right. So, this chip can help me to understand 3 different properties of the tissue. Now, if the tissue is from several stages, let us say benign, B-E-N-I-G-N, benign, or it is ductal carcinoma in situ, or it is invasive ductal carcinoma, or let us say it is ductal lobular carcinoma in situ, Lobular is cancer occurs in lobes, ductal is cancer occurring in ducts, and invasive lobular carcinoma. So, if I have tissue from several stages of the cancer, I can understand the change in the elasticity, resistance and thermal conductivity, and correlate that with my gold standard, and gold standard is the biomarkers that are available or that people use in the pathology lab. Oncopathologist will give us the understanding of whether the biomarkers are present, and with that study, if we correlate our electrical, mechanical and thermal property, can we add these modalities onto the existing modalities and can tell okay, the tissue is from this particular stage of the cancer. Thus, the electrical, thermal and mechanical property of a tissue we can measure with the help of this biochip, and if we are able to measure this and correlate with the gold standard, then we are helping to reduce false positive and false negative signals. You got it. So, the whole exercise of understanding the chip design, chip fabrication is to understand electrical, mechanical and thermal properties of tissue. So, it is very interesting, right, that by learning or by fabricating a biochip using a MEMS based technology, we can understand electrical, mechanical and thermal properties of tissue. Now, whether this thing works or not. So, as a part of research work which is published in IEEE Transactions on Biomedical Engineering, which I have given it to you right over here, right. We found that when you understand the mechanical properties, when you understand the electrical properties and when you understand the thermal properties of normal tissues and invasive ductal carcinoma, then there is a change in these three parameters. These are the SEM images. SEM stands for scanning electron microscopy, scanning electron microscopy. SEM images of scanning electron microscopy. You can also use FESEM, which is field emission scanning electron microscopy, field emission scanning electron scope microscopy. So, it is called FESEM. If you read, you will understand what is the difference between SEM and what difference between FESEM. Now, in terms of the morphology, in terms of the topography, what you can see? You can see that the normal tissues seems to be smoother compared to the carcinoma tissues, right. So, what does it mean? That means that if the topography is so rough, the resistance would be different compared to the normal tissue. If the resistance is different, the thermal conductivity should be different compared to the normal tissue, right. So, that what it means, you can clearly see that there is a delineation between IDCs and normal. Even you take a resistivity, the same thing, thermal conductivity is the same thing, right. So, you need to understand that this thing we can use it for measuring all three properties. Okay. Now, this is the blown up diagram that I was talking about little while ago, and you can see here there is this is made out of 3D printing, and we use object printing or object printer. Like I said, I will discuss about 3D printing in the in some other class. You can see that there are alignment columns, then there is a bottom part, a spring is there, microchip is there, a middle part is there, press fit contact pins are there, top part is there, and rubber packing is there, right. When you this is a blown up diagram, when you integrate all the things together, you can find out this particular casing, and this casing is with a biochip that is right over here, that we learn, right, the process flow during our other modules, and then these are the connectors that you can connect out, and you can understand different properties of tissue. So, this is an example. As I was talking, you can see an indenter. An indenter is attached with thermistor and electrode at the tip. So, the electrodes is used for applying the voltage between the top part of the or applying voltage across the tissue, and the thermistor is used to understand the change in temperature at the top surface when you heat the bottom surface, right. We discussed this thing. You can again connect this thing to a LCD to understand the different change in properties. Once you have a lot of data, you can also have the understanding where this tissue falls in which particular region, or it is from which particular stage. Now, the study was done using the dead tissues. That means the tissue is taken out, is freezed in the tissue bank, and as you can see, the paraffin is used to store the tissue. When we have to remove the paraffin, you have to do deparaffinization, that is done using chemical called xylene. Okay. So, paraffinized tissue, deparaffinized tissue. You can see the image here, photograph, where you can see breast tissue deparaffinized. This one is a breast tissue which is paraffin embedded, and once you have the tissue, you can place the tissue in this particular funnel, right. This whole thing is placed right over here, and the indenter is pressing the tissue. This we have discussed in the previous slide. All right. So, this is how the electrical, thermal, mechanical phenotyping of breast cancer we have seen. And one more thing that is interesting is that this system can not only help us to understand the change in the breast cancer tissue, but we can use this system for any tissue related cancer, or any cancer related to tissue based cancer, any tissue based cancer, that is the right term to use. Whether it is oral cancer, whether it is prostate cancer, even if we want to understand the stiffness of the gray matter, then also we can use the similar structure. What is the change in the stiffness of the gray matter? If a person has Alzheimer, a person is normal, you can see the change in the stiffness. Because stiffness is one of the properties of tissue that many people have used to understand the disease progression. All right. So, this is the last slide for this particular module, and what we have seen is that a chip that is using a MEMS based technology can be a chip fabricated can be used for understanding different tissue properties and for tissue related cancers like oral cancer, breast cancer. Here we have taken example of a breast cancer, but the same thing goes for the oral cancer. You can place the tissue on the chip and you can understand the change in the tissue properties. Now, so what does we saw? We saw that we just had only a oxidized silicon wafer, and from oxidized silicon wafer that we were using as a substrate, right, or in fact silicon wafer that we are using as substrate, we have grown oxide. On this we have deposited a heater, of pattern a heater, then we had an insulator, interdigital electrodes, piezoresistive sensor, insulator, gold pad, SU8 pillar, make it conductive. On the back side have a diaphragm. So you have all three sensors integrated onto this chip. When you slice the chip, when you slice this chip, what will happen, that you will have individual chips as I am showing it to you right here, right. Yesterday also we have last lecture, I do not know when I recorded, probably yesterday. So the point is, this is the chip that we obtained at the end of the process, and this chip is what we are using for understanding the change in the tissue properties. Alright guys, in next module we will discuss how can we just understand the change in the tissue properties which is mechanical properties. If I do not want to have this complicated structure, complicated sensor, because it requires several masks, how can I design something which is just for mechanical understanding. It depends. If you are designing a piezoresistive microcantilever, there are about 7 mask process. So depends. I am not talking about really the mask design, I am talking about the application point of view. If you want to understand only the stiffness of a cell, or you want to understand only stiffness of a tissue, why to go for electrical sensor and thermal sensors, right. So we will see in the next module how can you design a piezoresistive microcantilever that can be used to understand the change in the stiffness of the tissue. Again same thing, when I say tissue, it can be oral cancer, it can be breast cancer, it can be prostate cancer, it can be another tissue related cancer, but the focus will be on how can we fabricate. We will see the process flow, and we will also see what kind of mechanical properties we can measure with the help of piezoresistive sensor. Alright. So till then you take care, I will see you in the next module. Bye.